Hello everyone, my name is Ruiyan. My supervisor is Dr. Huang. The title of my presentation is Genomic Sequences and RNA binding proteins predict RNA splicing kinetics in various single-cell contexts. In this title, there are two important parts. First is the genomic sequences and RNA binding proteins, which are X values in our model. Second is the RNA splicing kinetics, which are Y value in our model. I will give this talk in four parts. First is the splicing efficiency. Second is the genomic features. The third part is the important features. In the last part, I will talk about the fit of our model in different tissues and species. First, let's look at what is the splicing kinetics and how can we quantify the kinetic rates. As we all know, DNA can be transcribed to the premature RNA. Then the premature RNA can be spliced into the mature RNA. Finally, it will be degraded. In this process, there are three kinetic rates. Alpha is the transcription rate, beta is the splicing rate, and gamma is the degradation rate. These two equations can describe this process. The unspliced RNA can be produced at a given transcription rate, and it will be spliced at a given splicing rate. The spliced RNA can be produced by the unspliced RNA at a given splicing rate, and it will be degraded at a given degradation rate. In these two equations, we always can obtain the abundance of unspliced RNA and spliced RNA. However, the transcription, splicing, and degradation rate are unknowns. So three unknowns are covered by two equations. How can we solve this problem? Here are some methods to answer this question. These approaches can be classified into bulk level and single-cell level. Some techniques such as TT-seq and SCNT-seq use the RNA metabolic label to obtain nascent RNA profiling and then measure the transcription rate. This greatly facilitates the resolution of the equation mentioned above. Another method is based on the mathematical modeling. They all have two solutions. First is based on the steady state observation. It relies on the strong assumption. And this assumption is the rate of splicing is constant. However, it can only calculate the ratio between the degradation rate and the splicing rate. Second is based on the time course data. Now, let's pay much attention on the ICV law, which would be main method used in our study. ICV law is a very useful extension of velocity, which first proposed the concept of RNA velocity. Velocity first infers the steady state ratio and then determines the velocity as the deviation of the observed ratio from the steady state ratio. In this process, velocity make a very important assumption. That is, all genes share a common splicing rate on the cellular level. However, this assumption is not always right, especially when a population includes multiple subpopulations with different kinetics. To solve this restriction, ICV law uses a expectation maximization framework to infer gene-specific kinetic rates. It defines this model as a dynamic model. At the same time, it also keeps a steady state model. In addition, It as a statistic model which exploits not only the relationship between unspliced and spliced mRNA abundance, but also their covariation. Our study mainly uses ISMB to estimate the kinetic rates. So we first explore the relationship between the splicing rate in dynamic model and the degradation rate in statistic model. We found that they are highly correlated. In addition, the degradation rate in statistical model can be mainly explained by the splicing rate in dynamic model. So we decided to choose the statistical model to do the next study because the statistical model is much simpler. Here, we define the relative splicing efficiency as a y-value in our model. It equals to the ratio of a splicing rate and the degradation rate. It also can be obtained directly from the ratio of abundance of spliced RNA and unspliced RNA. To prove the reliability of our y-value estimated from the ISMB law, we compare it with the y-value estimated from the velocity and the inspect manners, and we found that they are all highly correlated. In addition, we also found the relative splicing efficiency between the single cell estimated from the ISMB law and the bulk estimated from the inspect manners has a good match in K562 cell line. All of this evidence proves the accuracy of the y-value. Next, I will tell you what features are used in our study and how these features can be predict the splicing efficiency. This picture shows features used in our study, and these features can be divided into three groups. First is a basic feature set, including the splice site motif score, intron length, conservation of sequence, second structure energy, and the frequency of chimeras. Second is the octamer feature set, and third is the RNA binding protein feature set. All of these features can be fit into the random forest regression to predict the relative splicing efficiency. From this picture, we can see our model has a good fit in K562 cell line. Besides, we also explore the predictive ability of three feature sets mentioned above and found that RNA binding protein features are the most predictive one. In addition, the combination of two or three feature sets perform better than any set alone. This suggests each feature set contains independent predictive power. Our model can not only fit the splicing efficiency from the ISMB law, it also has a good performance on the y-value estimated from the TT-seq with the metabolic label and the RNA-seq without the metabolic label. To confirm the reliability of our model, we examined the non-setting by permuting genes and found that our model cannot predict the splicing efficiency at all. All of this evidence prove the good performance and reliability of our model. Next, we ask which features are important in this prediction. We rank these features according to their importance in the prediction and found that PABPC4 plays the most important role. The PABPC4 has been reported to be associated with increased splicing efficiency. In addition, we also zoomed into the prediction model with only one individual feature set and detect the important feature in the basic feature set. The importance of intron length is higher than others. In the RNA binding protein feature set, of course, the PABPC4 is the most important. In the octamer feature set, these two octamers play a very important role. Here, we display the relationship between the selected important features and the splicing efficiency. They are all highly correlated with our Y value. Because of the relationship between the RNA binding protein and octamers, we plot the relationship heat map of density of RNA binding protein and frequency of octamers. We can see the whole picture can be clustered into two blocks in the column, which indicates a negative correlation in the left and a positive correlation in the right. The ZNF800 has the strongest positive correlation with this octamer. And this octamer has a negative correlation with most RNA binding protein, which may be the reason why it is important in the prediction. Finally, we ask whether the genomic regulatory pattern also can be observed in other tissues and species. To answer this question, we first use the ICVelo to estimate the splicing efficiency on 5 tissues, including pancreas, dentity, gyrus, PBMC, and lung in mouse and human. Then we did correlation between them. We found that the intra-species pairs have higher correlation than the interspecies pairs. That may be due to the evolutionary divergence. Then we further use our model to predict the splicing efficiency from tissue mentioned above. We found that our model has a good performance on all of them. Finally, we wonder what is the extent of the regulatory of kinetic rates sharing across tissues and species. So we fit the regulation model from one tissue and predict the splicing efficiency on another tissue. We found that if a tissue cannot predict by itself, such as the dentity gyrus, it also cannot be improved by model fit from other tissues. Now, I would like to sum up our study. We first estimate the relative splicing efficiency by the statistical model in ICVelo. Second, we extract 3 feature sets, including basic genomic sequence features, octamer features, and RNA binding protein features to predict the splicing efficiency. Third, we identify the important features with strong regulatory potentials on splicing efficiency. Finally, we detect the regulatory pattern of kinetics rates across tissues and species. That's all I have to report. Thank you very much for your attention.